questions on Slido, but you're encouraged to continue to submit questions. And we already have a first one here. It says, could you elaborate on the technological advancements that have made FUS viable for epilepsy treatment and the anticipated future improvements? So, Dr. Wei, would you like to?
So, Dr. Yu?
So, I'm not really understand what the question is.
We also have a monitor in front, but it might be a bit small for the font.
Technology advance.
Okay. So far, with my limited experience in the clinical trial, it is a novel invention, but there are still a lot of improvements need to be done. For example, in my way, it's very clinical way thinking that the hair shaving, if the new transducer allow us to do, but we are still trying to do that. In the first pilot study, we shaved the scalp very, very meticulously, and my neurosurgeon makes some wounds on the scalp. But in the second trial, we started to leave some hair, and which is quite safe for us. So I think that sooner or later, we will have a scenario that we don't have to shave the hair to apply these treatments. And another way to improve, I think that the arm to fix the treatment is also very important. As in addition to NaviFast, I have seen other systems using the navigation, but a very useful arm like ROSA, robot-assisted arm, are also very useful for us clinicians in the future to do the treatment.
And Dr. O'Brien, also you have some experience?
Yeah, no, I would agree also with Dr. Yu about the hair shaving. Sounds like a simple thing, but certainly not so much for men like me who are hair shaved anyhow, but for a lot of our women it has been a barrier to recruitment, so I think if we could minimise that, that would help. I was going to comment, I think you can talk about the technical advances in the actual machine itself and the targeting and be able to observe real time the delivery of the treatment. And I saw some very nice technical advances when I was showing around the lab yesterday. But I think the real key thing is actually to work out the who and the where and the how in terms of which patients are best for this sort of treatment. And in my mind, this would be patients with well-defined epileptic foci that you can identify and target. Mesial temporal, as I said, there's a question why the hippocampus. Well, it depends where the epileptic zone is. That's the region of the brain that generates the seizures that you need to modulate to improve the seizures. And for the patients with mesial temporal lobe epilepsy, the hippocampus is the primary structure for that, which is why LIT, a very targeted laser ablation of hippocampus, can stop seizures in about 60% of people. So that's a very obvious group of patients to target. But really, theoretically, any patient with epilepsy who you can identify accurately the epileptic zone is amenable to this treatment. Then the other really important question is how much of that epileptic zone you need to treat. And I said in my talk, I think one of the mistakes we've made so far is we're only targeting a small region of the epileptic zone in the hippocampus. And I think if we target a larger region, we'll probably get better efficacy. So that's another advancement, I think. And then also, how often do we need to treat? How long do we need to treat for? And I would be very surprised, since this is not an ablation treatment, it's a neuromodulation treatment, I'd be surprised if it has a forever benefit from just one course of treatment. I would assume you'll need to repeat the treatment over time, as you do with most neuromodulating treatments. So how many treatments do you need to give each time, and how often do you need to repeat it? This is what good clinical studies will help us inform.
Okay, should we move on to the next question? The next question is for Dr. Ko-Ting Chen. Do you think the final results would be changed by large patient groups? What are the other side beneficial effects that could support the outcomes?
I did not mention about the clinical outcome of our phase 2 trial. However, our outcome is quite good compared to literature, but I think it's too limited number, so we definitely need a larger cohort. And I personally definitely hope that the outcome is not going to change. And for supporting that idea, we have the player signaling normalization, and also by using our system, we found that the elevation of cfDNA is correlates with the monitoring feedback system. That makes us more reliable or confident on what we are doing. One thing to mention is that from the literature, the recurrent GBM patients, the response rate is not homogeneous to bevacizumab. So after all, if we completed the trial, I think we can also study what is the best responder to the combination of FAS and bevacizumab with our trial.
Thank you. And our next question, should we? It's why choose the hippocampus as the target site to reduce the seizure activities?
Okay, let's go to the neurologist.
Campus? Yeah, because mesotemporal lobe, the MTS is the most common in adult patients with drug-resistant epilepsy, so it's easier to recruit patients for the clinical trial. And also, you see the DBS trial, the central trials, in the beginning, in the early analysis, that you can see the response of the temporal lobe epilepsy will respond better. So to include this group of patients, certainly you are supposed to target on the hippocampus. That's why we think hippocampus will be the target for neuromodulation in this group of patients.
Nothing more to add, right?
Thank you.
Thank you. Okay, the next question is for Dr. Rosian Chen. How does LIFU specifically modulate neural circuits in Parkinson's disease, and what are the underlying mechanisms that contribute to its therapeutic effects?
Okay, thank you for asking. But however, the mechanism is fully understood still right now. But when we talk about the neuromodulation hypothesis or mechanism, we all trap into the TMS theory. So we think my hypothesis that we can talk about the weak intensity or high intensity. I believe the weak intensity should through the mechanical channel, mechanical sensitive ion channel. For example, the most significant channel will be the piezo channel and the ascension ion channel and the transient receptor potential channel. The very low intensity will vibrate this bipolar layer and induce some cation channel changes. And the other is the high intensity, which is through the TBS or RTMS. This concept that will directly activate or suppress the direct wave of interwave. Therefore, in current publications, some research team, they use the same concept to do the study, but not the weak. So I think there is the weak intensity and high intensity. And if the high intensity comes, the weak intensity mechanism will be suppressed. That's my hypothesis. So the most important effect in the task is will be to settle down the protocol of tasks. For For example, TMS, we have the RTMS 1 Hz or 5 Hz. We have TBS, ITBS or CTBS. We have the PAS between the 5 milliseconds or 12 seconds. We have TDS patterns, but we still do not have the task patterns. So the first important issue is to identify and settle down the task protocol, and then to clearly identify the after effect. The after effect determinator should put some insight on the mechanism of the task. Thank you.
Thank you. And we're receiving a lot more new questions from the audience, and I think we will like to open later on 10 minutes for the audience to raise their hand and ask the questions, because there's just a lot. But I'm seeing there's one question directed to Dr. Musa right now. It says, if we scroll down a bit, there's one for Dr. Musa. It says, have you seen tumor regression on non-treated tumor and witnessed abscopal effects? Yeah, that one.
Yeah, that's a very good question. I showed an image from our fourth patient that was treated, and actually we saw a treatment effect in the area that was covered by ultrasound, but not in the uncovered area. So, at least in our 4 patients that we've done so far, we haven't seen tumor regression in the non-treated area. However, we are still in the process of analyzing the immunologic changes that occur. It is possible that we may see that, but so far we have not.
Thank you very much. And Dr. Wei, shall we now open the floor for questions in the audience?
So, is there any question from the audience?
So we have a lot more questions on Slido, and if you would like to now raise your hand and ask questions, we have our panelists here on the stage. Please feel free to raise your hand. Okay, we have one in the middle. Almost the same time. Thank you. You will kindly pass the microphone. Thank you.
Thank you very much for all those very nice presentation and studies. So for epilepsy, you showed that in the sham groups, there was some sort of spontaneous reduction in certain patients, sometimes really significant, but also with a huge variability from one patient to another. How do you think this variability could affect the group having first focused ultrasound stimulation?
Okay. Do you mean that, how do I explain the sham effect?
No, how this sham effect that you see, which is present in, I guess, in the group of patients, could affect the group that you include in the focused ultrasound stimulation arm.
There are 12 cases, so all the sham group were also in the final analysis. But in the first graph you see, I put all 12 cases together to see the 4 weeks of the sham response. So among them, some have already underwent focused ultrasound treatments 1 month before they receive focused ultrasound. So that's why I said there might be carryover effect from the previous focused ultrasound treatments. And also, it is very common to see almost 20% of the drug-resistant patients who enter the drug trial, new anti-seizure medication trial or new device trials. The placebo effect is very common, and sometimes in the early clinical trial, if the sample size is not enough, the response of placebo could be better than the target treatment.
Just to amplify that, Dr. is right. The epilepsy is inherently a fluctuating and random condition. Seizures they occur in clusters and they go for periods without them. Patients have good periods and bad periods, no matter what you do to them. So 20 to 30% of patients in drug trials have a 50% response on placebo. That's not because they're making it up, but because that's just an effect of the random fluctuation of seizures. So you're always going to see in sham studies some patients who seem to get better. just because of pure random chance, which does ultimately mean the importance of having randomised double-blind controlled trials to demonstrate efficacy in epilepsy. But at this point of the development of the FUS technology, we're really looking to try and explore what are the optimum treatment approaches, targets, patients, etc. And so that is actually best done in an open label environment. We have to understand the limitations of the data in that situation because of just the random fluctuations in epilepsy.
All right, and I've also received a question to Dr. Arian. Do drug uncaring face regulatory hurdles and challenging in the future?
Yes. So there's for the regulatory aspects, there's about the drug and then the device. So I mean, for the nanoparticulate formulation, there's a whole host of regulatory hurdles that get to overcome in developing a new injectable drug product. I'll just say that between the two vehicles that we developed, the liposomal vehicle is there's a lot lower regulatory hurdles about that because we're just slightly modifying a drug delivery vehicle that's fairly commonly utilized. And so the ketamine, clinical trial has gone, much faster than the propofol one, for instance, because of that reason. And then on the device side, on the ultrasound, so we've noticed that, if we provide more ultrasound intensity, whether duty cycle or pressure or both, we see more drug release. So and so we're actually intending to go past that magic MI 1.9, limitation that's there for diagnostic ultrasound, even though that we don't have a limit there for therapeutic ultrasound necessarily. So we are consciously going past that in our planned trial, but then we have to validate that in GLP safety toxicological studies in order to get there. So that is another hurdle because we have no actual guidance for therapeutic ultrasound about what's perfectly safe.
Thank you, and due to the time limitation, we can take one or two more questions from the audience. I believe we have one already. Thank you. Please pass the microphone.
Thank you. I noticed all the stories for epilepsy. The parameters were the same: 30% duty cycle, repetition frequency 100 Hz. What was the rationale to pick those parameters?
Because the time is limited, I didn't introduce our animal trial, animal studies. Our pilot study, we used the parameter that has been investigated in a paper published in 2020. We used in the PBC animal models and use 5 different level of sonication, and to do another simulation to test the temperature rising, and that's why we choose the 2.8 volts to be the target in the first pilot trial. And all the duty cycle and all the pulse rate frequency are all based on that animal studies.
Any panelists would like to add to that? And do we have any? Yes, we have one questions from the audience. Welcome.
My question is actually in a second. Our group has been doing ultrasound-induced apoptosis studies. So based on our experience, we can only detect apoptotic cells within at most maybe 2 days. So you said that you treated your patients like 2 weeks. You took the sample like weeks after the treatment, and yet you detected increased level of caspase activities. So what is causing these activities?
Yeah, that's a very good question, and I think it's something we don't fully understand. Interestingly, we did see increased cleaved caspase-3 in all 4 patients. So, if we did see some variability, I wouldn't believe it, but that does give some support to it. Yeah, I mean, it does suggest that there's increased apoptosis, 1 to 2 weeks after the resection. Now, I do have to admit there's also high variability in glioblastoma as well, so it's possible that this is not a real effect that we're seeing. So we'll have to see. And like I said in my presentation, it's still very early, only 4 patients have been treated, so let's see how. Thanks for your question.
Thank you. Do we have any? Okay, we have one more question from the audience. Right in the middle. Welcome.
Question for Dr. Yu. What is your speculation about the delayed response that you observed clinically from the FAST treatment?
Thank you for the question. Do you have any idea? I was thinking about how to explain this. Before, I don't believe that why we designed the clinical trial in 4 weeks, because I think that will be last only 1 or 2 weeks effect. But I didn't expect we can see that. But also in Dr. O'Brien's data, there are 2 cases. Seems that the response of residual reduction not occur as early as we expected. I have no good explanation on this because it's basically mechanical or thermal effect. How can it be modulated so delayed and lasted for months just after 1 shot of the treatment? So I don't know.
We have Dr. Aaron. Then Dr. O'Brien.
This is highly speculative, but I'm interested in just throwing something out there right before dinner. So this lymphatic result of ours, we see it's highly robust, and with a 10-minute focused ultrasound treatment one time, we can clear over half the hemorrhage from a mouse model of intracranial hemorrhage. And so we see it across a range of studies. We did look, and I guess there's at least some theory of a lymphatic hypothesis of epilepsy, that there's a lot of ECM buildup of junk, and exosomes and so on there. So I'll throw that out there.
Yeah, it's a really good thought. We're actually going to look at this, at least indirectly. Our director of neuroradiology, who's known to people here like Arthur, Professor Meng Law, has a particular interest in perivascular spaces, quantitative perivascular spaces, which are a measure of lymphatics. And so it's designed the MR that we've done pulled before and 3 months later after the ultrasound so that we can measure the lymphatics. So he and our research scientists are actually analysing the images to look at that.
Let's compare notes.
So it'd be interesting. But look, I just want to also step back a bit and just amplify what we said in our last discussion. Epilepsy is an inherently random, seizures in people with epilepsy are inherently random, unpredictable, they go up and down. So we just really need to be very cautious about over-interpreting any ups and downs with such small data in an open label study. It's interesting little signal. We've got some potential theoretical mechanisms, but we still need to get the fullness of data in more patients and ultimately blinded controlled studies before we interpret too much. Just one more thing to say, I like that microphone, is this delayed effect of an intervention in epilepsy is actually very well described. It's quite common in patients who have epileptic surgery, temporal lobectomy, to have some seizures in the first periods after the surgery, and then for them to wind down and become seizure-free long term. We do surgeries for patients with hypothalamic hematomas, which are hematomas deep in the brain. You see very active EEGs, lots of discharges all over the brain. When you ablate that hematoma, doesn't immediately those discharges stop, you find they wind down over months. So this idea that a neuromodulating intervention could have a slow, a impact that accumulates over months is actually quite well established in epilepsy.
If I may just add to that, if you look at the literature for responsive neurostimulation, you see better results in years down the line. So I think there are neuroplastic effects that occur, which may be the case here. I would expect that if you follow the data months, years down the line, it improves.
Okay, so before I turn the floor over to Dr. Wei for closing remarks, do our panelists have anything to add on? If not, Dr. Wei.
Thank you. For today's topic, for the brain tumor, for the seizure, for the Parkinson, and actually the most is exciting project is from the Dr. Wisha. The sonar dynamics. It is a very attractive point because if the sonar dynamic idea is come true, and then when we follow up the brain tumor, any new lesion maybe can control by the sonar dynamic therapy without the surgery. So hopefully we can go fast. This project in Taiwan, we will apply the sonar dynamic project very quickly. Hopefully we can get approval from the TFDA. And also the ultrasound is, I think the cutting edge of the ultrasound for the clinical application is quite quickly. Many diseases even from the BBB open, neuromodulation, even the sonar dynamic. So it's very congratulations for the Nevifoods company build up this very easy to use equipment, and then hopefully we can go fast.